Welcome to foodfeedinfo.com. Today lecture is about gluconeogenesis. Gluconeogenesis is the synthesis of glucose from non-carbohydrate stuff. It might be lactic acid, it might be amino acid, it might be fatty acid. So that depends on the situation. So in this lecture we will discuss about gluconeogenesis.
So question arises why gluconeogenesis is of vital importance or why it is required. If we look at different tissues like nervous tissue, erythrocytes, muscle, adipose tissue, mammary gland and fetus, all these require glucose as a source of energy. So the continual supply of glucose is only possible if there is a gluconeogenesis. Reason being, if diet is deficient or is lacking energy, then gluconeogenesis is the major pathway that can supply energy in the form of glucose. Maintenance of blood glucose level. Some cells and tissue, particularly brain and erythrocytes, they depend on glucose because they cannot oxidize alternate fuels. If we consider the human brain, it alone consumes 100 gram of glucose per day. Therefore, the body maintain a blood glucose level of 70 to 100 milligram per deciliter all the time, and this maintenance is particularly through dietary carbohydrates. So after a meal, the blood glucose level remain at a higher level for a few hours, and during these hours the blood glucose concentration can rise as high as 150 milligram per deciliter. But during the fasting state, there is a deficiency of glucose, then body have to produce the glucose to make these organ or tissues or cells in a functioning state to provide the fuel to these cells and tissues. So in the fasting state, the liver has to produce the glucose by two pathways. The pathways are either glycogen degradation or the second one is the gluconeogenesis. We will come to this point. When we compare human or monogastric species with the ruminant animals, in case of ruminant animals, the ruminant nervous tissues, they use 600 gram of glucose per day, and they can tolerate a long period of hypoglycemia. So this is the major difference between ruminant and non-ruminant animals, or monogastric and chambered stomach animals. So they can maintain or they can survive even a low glucose level of 18 milligram per deciliter for up to 6 hours without having any deleterious effect on their body functioning. So this is a major difference. And the reason, the most probable reason is the brain and spinal cord of ruminant animals is tend to be lower as a percentage of body weight as compared to the monogastric animals. So now we will look at the options, how this gluconeogenesis is going on, or it can be required for muscles and adipose tissues. In the previous slide we have discussed the nervous tissue or brain. So when we look at the muscles, they require glucose primarily to produce glycogen, although a small amount of glucose is oxidized directly. So when we compare the ruminant muscle, they contain much less glycogen than that of other species, so it reflects the lower amount of glucose available at the tissue level. In adipose tissues, glucose is required as a source of glycerol. So it provides the glycerol for the synthesis of triglyceride, and it is probably it play a major role in maintaining the high level of intermediate of the citric acid cycle in many tissues. Glucose is also required for the fat metabolism because it furnishes NADPH, which is formed by glucose oxidation via the hexose monophosphate pathway, and NADPH is required as a reducing agent at the recurring steps in the synthesis of long chain fatty acids. So for adipose tissues, this glucose is important because it provides NADPH through hexose monophosphate pathway, and NADPH is utilized as a reducing agent during the synthesis of long chain fatty acids. When we talk about the fetus, here is the example of sheep. Its fetal glucose metabolism, it accounts for 40 to 70% of the total glucose metabolized in the body. And when we take an example of milk, so milk contains 5% lactose, so if there is almost 2 kg glucose per day is the requirement for 40 kg of milk production. So if an animal is producing 40 liter of milk, and if we consider the lactose as a 5%, so there is a requirement of 2 kg glucose per day for that animal. When we talk about high producer animal, definitely the lactose production will be more, and it can account for 60 to 85% of the total body metabolism. So what are the options which are available to maintain the blood glucose level, particularly during the fasting state? Because we all know that just after a meal the blood glucose level is usually high, and it can go up to 150 milligram per deciliter, and during a fasting state the blood glucose level is low, and body should undergo gluconeogenesis or some other pathway to supply the glucose. So it means in monogastric species the gluconeogenesis is at its peak during the fasting state. So this is entirely different from that of ruminant animals, because the blood glucose level is usually low in case of ruminant animals, and gluconeogenesis is at its peak just after the meal. The reason being the precursor for glucose synthesis, they are available just after the meal. So if we look at the supply of glucose during the fasting state, or the state when glucose level is low, the first one is glycogen degradation in the liver. The glycogen degradation is fast, and it's the cheapest source to supply the energy, so because it requires no metabolic energy. But the major issue is the glycogen reserves of the liver. They are very less. They usually you can say up to 100 gram, and therefore they are depleted within a very short time, and it can go maximum up to 24 hours if we talk about the human being. So only liver glycogen, but not the glycogen of the muscle and other tissue, undergo degradation, and they can be used to maintain the blood glucose level. The second and the major option is the gluconeogenesis, that produce glucose from amino acid, lactic acid, and glycerol. Gluconeogenesis is mainly occurred in case of liver and kidney. They both have a complete gluconeogenic pathway, but on a major basis the liver is the major glucogenic organ because of its large size. So gluconeogenesis is the only source of glucose during prolonged fasting state. So gluconeogenesis is the only source of glucose during a prolonged fasting state. So how we can define the gluconeogenesis? It is the synthesis of glucose from non-carbohydrate precursors. These precursors include glucogenic amino acid, glycerol, and lactate. And when we talk about the ruminant animal, propionate is the gluconeogenic source or precursor. However, a small amount of glucose it can be made from short chain fatty acids. Gluconeogenesis is the only source of glucose during prolonged fasting. This is what we are stating. So all the substrates which are used for gluconeogenesis, they enter the TCA cycle, and eventually they yield malate, phosphoenol pyruvate, and the glucose. Channels of gluconeogenesis. The first one is reverse glycolysis, and it need to bypass 3 irreversible steps of glycolysis. So 3 irreversible steps of glycolysis, they should be bypassed, or these steps are catalyzed by hexokinase, phosphofructokinase 1, and pyruvate kinase. The second is through lactate and alanine. Third one is oxaloacetate, and the fourth one is glycerol. So these are the 4 major channels of gluconeogenesis, and we will discuss one by one in the next coming slides.
Gluconeogenesis or reverse glycolysis. The first step in the gluconeogenesis is a conversion of pyruvate into phosphoenol pyruvate. Just keep in mind, even if there is a lactate, that should first be converted into pyruvate. So glucose cannot be produced from pyruvate by a simple reversal of glycolytic pathways, because there are the three reactions which are irreversible. The first is conversion of pyruvate to phosphoenol pyruvate, and then fructose 1,6 diphosphate or bisphosphate into fructose 6 phosphate, and third one is glucose 6 phosphate to glucose. So these are the three irreversible reactions. So in this slide we will see how this pyruvate is converted into phosphoenol pyruvate. If we look at a simple glycolytic pathway, the pyruvate is converted into either acetyl CoA or lactate, but if it has to be converted into phosphoenol pyruvate, so this is the first hurdle step, because pyruvate kinase cannot be reversed, and this reversion require an energy. During this reversion process, the pyruvate is converted into phosphoenol pyruvate through a sequence of two reactions. In the first reaction, pyruvate is converted into oxaloacetate with the help of pyruvate carboxylase. So this carboxylation is ATP dependent, and this reaction is or this enzyme is strictly a mitochondrial enzyme. So in the first step, pyruvate is carboxylated, and that result in the production of oxaloacetate, and there is a requirement of 1 ATP at this step. In the second reaction, the oxaloacetate is converted into phosphoenol pyruvate, and this reaction is catalyzed by phosphoenol pyruvate carboxykinase. Phosphoenol pyruvate carboxykinase it requires GTP. So it supplies the phosphate group in the phosphoenol pyruvate. These two reactions, when they are combined, they have a free energy change of almost 0.8 kilojoule per mole. So this is how this pyruvate is converted into phosphoenol pyruvate, and this complete conversion requires 1 ATP and 1 GTP. Because there is a requirement of 6 carbon molecules, so 2 pyruvates they are required to pass through these reactions. So it means in total 2 ATPs and 2 GTP they are required during this step, so that it can furnish 6 carbon glucose at the end. The pyruvate phosphoenol pyruvate carboxykinase, phosphoenol pyruvate carboxykinase of the gluconeogenesis is mainly cytoplasmic. So in the next slide we will see the next reversible or irreversible reactions, how gluconeogenesis proceed further. This is what we have discussed as a reverse glycolysis. From pyruvate there is a production of oxaloacetate and then phosphoenol pyruvate, that is then converted into 2-phosphoglycerate, and then it is converted into 3-phosphoglycerate. So these are the reversible reaction, and they do not require any change in the enzyme or use of the energy. Then 3-phosphoglycerate is converted into 1,3-bisphosphoglycerate. This step require again ATP to convert it into 1,3-bisphosphoglycerate. And then for the next step, for the formation of glyceraldehyde 3-phosphate, there is a utilization of again NADH to produce glyceraldehyde 3-phosphate. So coming back to the step of this production of this oxaloacetate and then phosphoenol pyruvate, the second reaction which is carried out by phosphoenol pyruvate carboxylase, which convert oxaloacetate to phosphoenol pyruvate, may take place in the mitochondria, and the phosphoenol pyruvate then passes into the cell cytoplasm. The alternate option is oxaloacetate. It may be transferred into the cytoplasm by means of glutamate aspartate shuttle and converted to phosphoenol pyruvate by cytoplasmic phosphoenol pyruvate carboxylase. So when we compare it with the ruminant animal, pyruvate carboxylase is located in the cell cytoplasm as well as the mitochondria, and pyruvate can be changed to phosphoenol pyruvate entirely in the cytoplasm. So at the end there is a production of glyceraldehyde 3-phosphate, and if we look at this glyceraldehyde phosphate, it is interchangeable with dihydroxyacetone phosphate and they are converted into fructose 1,6-bisphosphate. Now this reaction is irreversible. Fructose 1,6-bisphosphate cannot be converted into fructose 6-phosphate until there is fructose 1,6-bisphosphatase is there. So that result in the production of fructose 6-phosphate, and also there is a production of inorganic phosphorus. Then again the reversible reaction, fructose 6 is converted into glucose 6-phosphate as in case of glycolysis. Then again the third irreversible reaction is conversion of glucose 6-phosphate into glucose, and it can be accomplished by glucose 6-phosphatase with the production of an inorganic phosphate. So three reactions which are irreversible. The first is glucose 6-phosphate conversion into glucose, second fructose 1,6-bisphosphate conversion into fructose 6-phosphate, and the third one is production of phosphoenol pyruvate from the pyruvate. So pyruvate is first converted into oxaloacetate, then into phosphoenol pyruvate. So if we look at the energies of this gluconeogenesis, so this reaction or the complete accomplishment of glucose synthesis requires 6-phosphate bond in the form of ATP GTP. Now we will talk about the energy expenses during the formation of glucose from pyruvate or lactate. So in generally we can say gluconeogenesis is energetically expensive but an essential process. So if we look at the reactions and we sum up these reactions, the formation of glucose require either 2 pyruvate or 2 lactate. So we can say total there is a requirement of 2 pyruvate and then 4 ATPs, 2 GTP, 2 NADH and 2 hydrogen and 4 water molecules. The first 2 ATPs they are from this pyruvate carboxylase, and then 2 GTP from pyruvate phosphoenol pyruvate carboxykinase, then from phosphoglycerate kinase that also consumes the ATP. And all these three reactions they occur twice because we are depending on 2 pyruvate molecule for the synthesis of 6 carbon glucose structure. So for each molecule of glucose formed from pyruvate, 6 high energy phosphate groups are required, 4 in the form of ATP, 2 in the form of GTP. So in addition to these, 2 molecules of NADH are also required for the reduction of 2 molecules of 1,3-bisphosphoglycerate. So in total we can say there is a requirement of 4 ATP, 2 GTP and 2 NADH. So overall this is a very energy expensive process, but because there is a requirement and the body need glucose, particularly when there is a deficiency of glucose, so that's why body is going through this process. Even this is not considered as an energy efficient process, rather than an energy expensive process. Now come to the next glucose channel. We have discussed the reverse glycolysis, and the next one is the lactate or alanine. So lactate and alanine, they are convenient substrate as they are readily converted to pyruvate by lactate dehydrogenase or by transaminase respectively. So that's why they can be used as a source of gluconeogenesis or a precursor of gluconeogenesis. Then comes the oxaloacetate. Oxaloacetate is usually formed during the TCA cycle But oxaloacetate is not only a gluconeogenic intermediate but also a member of the TCA cycle. So this is important because most amino acids are degraded to TCA cycle intermediate. Through the TCA cycle, these glucogenic amino acids they feed into the gluconeogenesis. The last one is the glycerol, that is available from fat breakdown or lipolysis. So it enters the pathway at the level of triose phosphate. So we will see in the next slide. Glycerol enter the gluconeogenesis and we can say also the glycolysis at the level of triose phosphate. So now if you look at the glycerol molecule is converted into either glycerol 3-phosphate through glycerol kinase. It is converted into glycerol 3-phosphate. From where this glycerol is coming? It is coming from the adipose tissues. During the lipolysis, there is a formation of glycerol and free fatty acids. So glycerol, with the help of glycerol kinase, it is converted into glycerol 3-phosphate. So this reaction requires energy. Glycerol 3-phosphate then converted into dihydroxyacetone phosphate, or that can be interconverted into glyceraldehyde 3-phosphate. Again, the conversion of glycerol 3-phosphate into dihydroxyacetone phosphate is catalyzed by glycerol phosphate dehydrogenase, and there is also a production of NADH. So now this dihydroxyacetone phosphate or glyceraldehyde 3-phosphate, they can follow the pathway of glycolysis. Rather than being converted into glucose, they can undergo glycolytic pathway and then TCA cycle. But when there is a need for glucose by the brain or RBCs, or blood sugar level is low, then they are converted into fructose 1,6-bisphosphate, and they follow the two irreversible reaction for the conversion of fructose 1,6-bisphosphate into fructose 6-phosphate, and then fructose 6-phosphate into glucose 6-phosphate, and then glucose 6-phosphate into glucose. So these steps are similar, which we have discussed in the previous slide during this reverse glycolysis, and this glucose is then utilized by the cells or tissues. So this is how this glycerol is utilized for the. So this is we can say the summary of gluconeogenesis, or the most important substrate of gluconeogenesis. The biosynthetic pathway to glucose that we have discussed, its synthesis from glycerol, from lactate, from pyruvate, from oxaloacetate, or from alanine amino acids, that we all have covered. But keep in mind, the synthesis of glucose cannot occur only from pyruvate or the substrate which we have discussed, but it can also from the 4, 5 and 6 carbon intermediate of the citric acid cycle. For example, citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate and malate all are citric acid cycle intermediate that can undergo oxidation to oxaloacetate. So few of them, or all of the carbon items of most of the amino acid derived from the proteins, they are ultimately catabolized to pyruvate or to intermediate of the citric acid cycle. Such amino acids can therefore they undergo net conversion to glucose, and these amino acids are known as glucogenic amino acids. Glutamine and alanine, these are the principal molecules that transport amino group from extrahepatic tissues to the liver, and particularly important glucogenic amino acid in mammals. So after the removal of their amino group in the liver mitochondria, the carbon skeleton remaining are readily funded into the gluconeogenesis pathways, or you can say that they are then funneled or fueled into the gluconeogenesis pathway. So pyruvate and alpha-ketoglutarate, they are respectively are the cases in case of liver mitochondria, and they provide this structure. Now look at now look at this complete picture of gluconeogenesis, where I have tried to combine all glycolytic pathway and TCA cycle pathway and those reversible or irreversible reactions which plays a key role during the gluconeogenesis. So here you can see the glycerol is converted into triose phosphate, we have already discussed. Then the amino acid like alanine, glycine, serine, cysteine and the lactate, they are converted into pyruvate in the cytoplasm, and then this pyruvate is taken into the mitochondria and converted into oxaloacetate, phosphoenol pyruvate, and then taken back to the cytoplasm. Then from oxaloacetate converted into aspartate or malate shuttle, and then converted into again oxaloacetate in the cytoplasm, and then taken to or converted to the phosphoenol pyruvate. If we look at the TCA cycle, at alpha ketoglutarate level, glutamine, histidine, proline and arginine, they enter at this level, like valine, threonine, methionine and isoleucine, they are converted into succinyl CoA. Likewise, propionic acid, which is a VFAs produced during the ruminal fermentation, is converted into succinyl CoA. Then tyrosine and phenylalanine, they are converted into fumarate or fumaric acid. So this is what we say is the gluconeogenesis, or if we start from the glucose, is the glycolytic pathway. So how they are interlinked. But now my question is, where is the role of fat, particularly when we are obese and we think that during the deficiency of glucose this fat will be burned or will be utilized. So keep in mind fat cannot be a source of glucose, particularly when there is a deficiency of glucose. So if we look at the book, so in contrast, no net conversion of fatty acid to glucose occur in mammal. The catabolism of most fatty acid yield only acetyl CoA, and mammals cannot use acetyl CoA as a precursor of glucose. What is the reason? The pyruvate dehydrogenase reaction is irreversible, and cells have no other pathway to convert acetyl CoA to pyruvate. Plant, yeast and many bacteria do have a pathway, the glyoxylate cycle, for the conversion of acetyl CoA to oxaloacetate. So that's why these organisms can use fatty acid as the starting material for gluconeogenesis, and this is especially important during the germination of seedling before photosynthesis can serve as a source of glucose. So in case of animals, they cannot convert acetyl CoA derived from the fatty acid into glucose. So that's the reason when even with fat in the body, but that fat cannot be converted into glucose. So this is what I want to make it clear, particularly when we think that even if we have a fat, then why this happened. In case of ruminant animals, yes, acetyl CoA or acetate is utilized as a glucogenic precursor when there is a deficiency of glucose, but again in some cases due to the deficiency of citrate lyase, they wouldn't be able to synthesize fat, and that result in the accumulation of fatty acids in the form of ketone bodies. So when fat is mobilized, that result in the production of acetyl CoA, and citrate lyase is if it is not there, so that result in the accumulation of acetyl CoA, and that is converted into ketone bodies, acetoacetate, beta-hydroxybutyrate or acetone, and that result in ketosis in ruminant animals. So this is all about gluconeogenesis. I tried my best to convey the maximum information that I have or that I can for this gluconeogenesis. So if you have any question, you can comment or you can ask, and I will try to cover some aspects of regulation in some coming video. Thank you for watching.